In this session, we study statistical inference for linear regression analysis, with focus on p-values, confidence intervals, and assumptions which those computations are based on. In sessions 2 and 3, we studied basic models of linear regression, with focus on roles and interpretations of partial regression coefficients, alpha and betas. In session 4, we studied how those alpha and betas are estimated. Now in session 5, we study how reliable those estimates are, and how the reliability of those estimates are assessed. We will study computations of p-values and confidence intervals for linear regression analysis, mathematical assumptions on which those computations are based, and basic procedures of diagnostics of deviations from those assumptions. Topics of this session are organized as shown in the slide. In order to get some ideas about the effects of sampling variations on estimated values of regression coefficients, let's look at results of some computer simulations. In the simulations, it was assumed that relationships of systolic blood pressure to gender and age can be expressed by this model. In this model equation, Y is systolic blood pressure, x1 is the binary variable for gender, and x2 is adult age. It was assumed that the value of alpha is 100, beta 1 is 4.0, and beta 2 is 0.5. And epsilon, the residual, was assumed normally distributed, with a mean of 0 and the standard deviation of 13. Using this model and these parameter values, simulated values of y, x1, and x2 were produced for 200 hypothetical persons. Then a linear regression analysis was done, using this simulated data, and the following results were obtained. The estimated value of alpha was 100.516, which is very close to the true value of 100. The estimated value of beta 1 was 7.182, which is quite different from its true value of 4.0. Finally, beta 2 is 0.456, somewhat different but not very different from its true value of 0.5. This is a plot of y against y hat for the 200 hypothetical persons. Certainly y and y hat are noticeably correlated, but the points are scattered around the red line indicating y equals y hat, and many points do not appear very close to the red line. R squared is about 0.4. This spread of points in the plot gives us an idea about the extent of randomness in the regression analysis results. The extent of randomness in the simulated data was determined by the standard deviation of epsilon. After this simulation, two sets of 200 simulations were done, and let's call them Simulation 1 and Simulation 2. In Simulation 1, 200 data sets were produced using the same parameter values as those used in the simulation in the last few slides, and then 200 regression analysis was done, using each of the 200 datasets. In Simulation 2, the value of B1 was changed from 0.4 to 0.0, but the other parameter values were kept unchanged, and then another bunch of 200 datasets and regression results were produced. Note, the number 200 is used here for two completely different things. In each of the two simulation exercises, a set of 200 simulations were done, and each of the simulations produced artificial data on 200 hypothetical persons. These two graphs show the distribution of 200 values of B1 in Simulation 1. The left panel shows 200 vertical bars, indicating the 200 values of B1, from the lowest to the highest. The right panel is a histogram summarizing the distribution. The value of beta 1 is 4.0, so in this hypothetical population, men have, on average, 4-point higher systolic blood pressure than women, controlling for age. But its estimate ranged from -1.4 to 9.2. The highest value, 9.2, seems quite misleading, suggesting the average gender difference in systolic blood pressure is much greater than the true average difference of 4.0. And the lowest value of -1.4 seems even more misleading, because it suggests that men tend to have slightly lower systolic blood pressure than women, even though the truth about this hypothetical population is, men tend to have higher systolic blood pressure than women. These two highest and lowest estimates may be outliers, but more than 10% of B1s were lower than 1.0 or greater than 7.0, quite different from the true gender difference of 4.0. These two graphs are from Simulation 2. They are the same types of graphs as those in the previous slide, but in this simulation, there is no average difference in systolic blood pressure between men and women in this hypothetical population. Nevertheless, the value of B1 ranged from -4.1 to 5.5, whereas the true value is 0. More than 22 of the B1 values, which is more than 10% of 200 B1 values, were less than -3 or greater than 3. Values of A, B1, and B2 in simulations 1 and 2 are shown in the Excel file labeled simulations.statinf, which is included in the materials of this session. Note that in actual research projects, we never get these kinds of graphs. Our dataset in our regression analysis correspond to just one of those vertical bars in the left panel. If we are lucky, our result will be like a bar around the middle. If we are not lucky, our result may be like a bar near the right or left end of the graph, so quite different from the true value. For small studies, the sample size of 200 is not unusually small. So we should carefully assess the extent to which our results could be affected by sampling variations.